Well, it's a pleasure to be here, particularly because it's easy to come to a local meeting. So what I thought I would do today is to discuss why cancer is such a hard disease to treat and to cure. And I thought I'd begin... seem to have locked up somehow. Oh, I see. It's not changing on my screen down here. Okay, so that's a problem. So I thought I would start with, some definitions. The Greek physician Hippocrates created two terms to describe cancer: onkos, meaning a mass or swelling for benign tumors, and karkinos, meaning crab, for malignant tumors, because he thought the finger-like spreading projections from a cancer resembled a crab. And you can see that in this section of a breast cancer. You can see the threads of the tumor spreading out from a center here. And then the word tumor comes from the Latin word tumere, which means, again, to swell. Now, cancer is a disease, at least the first record we have of it was from the Egyptians over 4,000 years ago in their texts. An examination of a 4,000-year-old skull suggested some efforts have been made to try and treat a cancer. So cancer remains a scourge worldwide. 20 million cancers were diagnosed in 2024 with 10 million deaths, with lung cancer accounting for about 12% of all cancers and breast cancer around 11% of all cancer. In the US, the numbers are really similar: 2 million new cases last year with over 600,000 cancer deaths. And cancer, as we heard earlier in the meeting, is the second leading cause of death, just behind cardiovascular disease. And in terms of the frequency of cancers, you can see from the 2024 data that the most common cancers were breast, prostate, lung, and colorectal cancer. Now, there has been some success in reducing cancer mortality. As you can see, for the bell-shaped curve for lung cancer, there's been a significant drop over the last 30 years or so. This is largely due to smoking cessation and partly due to better treatments. There's been drops in colorectal cancer, probably due to colonoscopy diagnosis, and also in breast cancer, largely due to successful treatment. So we recognize three different types of cancer. Carcinomas, the most common cancers, derived from epithelial cells such as breast, lung, colorectal. Sarcomas derived from mesenchymal cells, bone and muscle. And leukemia and lymphoma derived from blood-producing cells. Now, one important step in understanding the disease and beginning to think about how to treat it was Nixon's war on cancer. He signed into law the National Cancer Act in December of 1971. This gave the director of the National Cancer Institute a number of powers, including creating new cancer centers, and the Salk Institute Cancer Center was the first cancer center to be designated in California in 1973. Now, another important effort by the NCI in the early days was to create a special virus cancer program. And this was important because many of the early efforts to try and understand cancer were through the use of model systems, tumor viruses that cause tumors in birds and rodents. And these have been used since the beginning, in fact, of the 20th century. And these viruses not only cause tumors in animals, but can also transform a normal cell in a culture dish into a malignant state. There are two main types of tumor virus. The DNA tumor viruses, who have a DNA genome, and these are from 5,000 to 150,000 base pairs that encode 5 to maybe 50 or so viral proteins, only some of which are needed for cell transformation. The others are structural proteins. And then the RNA tumor viruses, so-called retroviruses, have a single-stranded RNA genome that's copied into DNA to be integrated into the cellular genome, and these transform cells by having captured a cellular gene. As an example, you can see here normal cells on the left and RAS sarcoma virus transform cells on the right. And if you look at the scanning electron micrograph on the bottom, you can see how these cells have rounded up and become detached. Properties you would expect of a cancer cell. Now, we've known for many years that cancer has a genetic basis, and as a resulting from mutations in cellular genes. And in the 1950s, Richard Doll, the British epidemiologist, deduced, based on the shape of the age incidence curve, that 5 to 7 genetic changes would be needed to create a cancer. So that raises the question then of which of the 20,000 genes we have need to be mutated to cause cancer. And an important step in this direction was this perspective written and published by Renato Dulbecco, who was at the Salk Institute at that time, proposing that in order to understand the cancer genome and what changes had occurred, we would need to know the sequence of the normal human genome. And so he, in this thought piece, suggested we needed to sequence the human genome. And as you're all aware, that happened, and 15 years later, the completion of the human genome was announced. And then it became possible to compare that sequence to the sequence of the genome of a cancer cell. One of the early publications in this regard was the comparison of the sequence of a non-small cell lung carcinoma tumor and the normal DNA from a blood cell from the same, as it turned out, a smoker patient. There were a huge number of changes in the cancer cells. And you can see the arrow's bullet point there shows that there were over nearly 400 changes in the genome which would alter the nature of a protein, including the KRAS protein. So then the question is, which of this huge number of mutations were the driver mutations that actually caused this cancer, and which were silent or passenger mutations? Now, I should point out that lung cancer typically has a very high mutation burden, largely because of environmental exposure, such as to smoke. But melanoma, which also is exposed to UV light, which is a mutagen, which is why we use sunscreen, also has a very high mutation burden. But there are tumors like rhabdoid tumors on the right here, which have only a few, maybe just one mutation. And so there's a huge variation in the number of genetic changes in different types of cancer. We recognize two types of gene that are commonly mutated in cancer. There are oncogenes, derived from the word onkos, obviously, which you can consider as accelerators of cell growth. These genes have what we call gain of function activating changes that switches their activity on constitutively and drives cell proliferation. The second class are the tumor suppressor genes, whose function, as the name suggests, is that they actually suppress the growth of cells. But in this case, because we have two copies of every gene, both copies of that gene have to be inactivated by a mutation. Now, I'll just give you a couple of examples of the sort of genes that we call oncogenes, that are the tyrosine kinases that you heard about in the introduction. These are enzymes that add phosphate to proteins, particularly on the amino acid tyrosine. And they were discovered actually through studying tumor viruses. But then the subsequent discovery that there are tyrosine kinases in human cancers that are constitutively activated by mutation led to the development of this large number of tyrosine kinase inhibitor drugs. And then RAS, which I already mentioned, again discovered through work on tumor viruses. And here just a single change in the amino acid sequence causes constitutive activation. These were discovered in the early 1980s, I would say. Now, this protein was once deemed undruggable. For 50 years, we had no way of blocking its activity. But in the last few years, a new class of inhibitor drug has been approved for cancer therapy. And then tumor suppressor genes, TP53, sometimes known as the guardian of the genome. This protein triggers cells to commit suicide when it senses cellular damage that can't be repaired. It's a way of eliminating bad cells, including cancer cells. And interestingly, TP53 was also discovered through work on tumor viruses. And then finally, retinoblastoma. The loss of both copies of retinoblastoma causes, a retinoblastoma gene causes the eye tumor retinoblastoma. And in normal cells, retinoblastoma prevents unscheduled cell proliferation. The COSMIC database maintained at the Sanger Institute in Cambridge in England lists around 700 human cancer genes. These are genes that are consistently mutated in cancer cells of different types. Now, when we were working on discovering oncogenes, we set up an oncogene meeting, which was held for many years. And each year we would devise a cartoon to go on the abstract book cover. And in 1989, Jamie Simon and I devised the Oncopoly game, which you can see on the right-hand side are the tyrosine kinase oncogenes. On the bottom right, there's the RAS oncogenes, etc. They're actually arrayed in the order of their molecular mass. And, of course, there are chance cards. And it's a little hard for me to read here, but unauthorized,